Welcome to our lecture on mechanisms of infectious disease. So we have this microbial balance. Many microbes live on our skin and within our mucous membranes. We call this our normal flora. It helps with digestion of nutrients, to produce vitamin K, and to inhibit other bad bacterial growth. The Human Microbiome Project at the National Institute of Health has found that there's over 100 trillion bacteria on us. That's anywhere from 2 to 5 pounds. So congratulations, you're 2 to 5 pounds lighter than you thought you were. There's 1,000 strains identified by gene sequencing. These strains differ between people. Changes can lead to problems such as premature birth. Let's go over some terms that we commonly use. First is host. That's the supporting organism, and typically we're referring to ourselves. We're the host. Colonization is the presence of those thousands of microorganisms that are on us. An infection is the presence and multiplication and tissue injury that occurs from these microorganisms. Virulence is the potential to produce disease, and pathogens are virulent organisms. Saprophytes are harmless organisms. Opportunistic are ones that cause disease when somebody is immunocompromised. Prions are protein particles that lack a genome and are categorized as transmittable neurodegenerative diseases. A prion particle is resistant to enzymes which normally break down proteins. Replication is not fully understood. Now, normal cells are tricked into producing abnormal proteins which deposit as amyloid plaques. So let's go over the pathophysiology of a prion. It's a slowly progressive degeneration of the nerves. We get amyloid plaques that deposit in the brain and spread throughout the nervous system. Prion proteins are mutant forms of normal host proteins which cause this disease. Some examples include chronic wasting disease, bovine spongiform encephalopathy, and symptoms include ataxia, so difficulty or abnormal movement, dementia, and wasting. Most prions do not affect humans. Let's move on to viruses. So, a virus is a small intracellular pathogen. They thrive and reproduce only in a host's cells. So they're simple organisms and they have no organelles. They use our organelles to reproduce. The basic structure is the viron, and it contains nucleic acid protected by a capsid, which is a protein capsule. Some are enclosed with a lipoprotein envelope, and there are 7 classes based on the presence of DNA or RNA, and if it's single or double-stranded. The viral genome can be either an RNA or a DNA, and it can be single or double-stranded. It uses retrovirus replication, and the disease can produce things like hepatitis A, B, and C. So the life cycle of a virus includes attachment of the virus to the cell surface protein receptors. It then penetrates that host cells. It uncoats itself and releases that nucleic acid, and then replication occurs. Assembly of a new virus, and then the release of that new viron into the system. We do have some defenses against viruses though. We have antibodies, cellular immunity, and interferons. Viruses can cause damage to the cells because it disrupts the normal protein synthesis within the cell. As a result, it promotes apoptosis of that cell. It can also change the cell and cause those infected cells to become malignant. So the influenza virus has 2 surface proteins, the hemagglutinin H and neuraminidase N. So H and N. You may have heard of swine flu, which is H1N1. So there's an antigenic shift of surface proteins that can occur yearly. So you can have H1N1, you can have H5N1, and so that's how the flu viruses shift. We can also develop an acquired immunodeficiency syndrome. It can be transmitted sexually, that's the most common way. It can be perinatal, it could be through breast milk, or through blood or blood products. This is when we have our CD4 and T cells that get infected, leading to infection and malignancy. The genetic information is carried in RNA, and it reproduces through enzyme reverse transcriptase, which converts that RNA to double-stranded DNA. Here's an example of human immunodeficiency virus structure. We're going to go over the stages and then treatment of an HIV infection. So the primary infection is stage 0. This is in the first 180 days. Then you're going to have the latent or stage 1 or stage 2 based on the patient's CD4 counts. This is when a patient has a chronic and often asymptomatic response. AIDS will eventually develop at stage 3. This is when it's acquired immunodeficiency that came from that HIV virus. Stage 3 is based on those CD4 counts or development of AIDS-related opportunistic infections. Treatment is through highly active antiretroviral therapy, or HAART. Another virus is the Zika virus. This is transmitted through mosquito bites, could be perinatal, sexual, or through transfusions. Symptoms are often asymptomatic. It includes fever, rash, arthralgia, conjunctivitis, headaches, and myalgia. Complications include Guillain-Barré syndrome, fetal brain defects such as microcephaly, so a small brain. The way we diagnose these patients are through doing clinical signs and symptoms and finding out if they've been exposed to it. We can also do labs for the virus and IgM. There are different types of bacteria out there as well. We have prokaryotic. We can have prokaryotic unicellular, one chromosome plasmids with circular DNA. And categories are based on morphology, so how they look, and staining. So we can put stain in there and see what color they come up. So they could be cocci or bacilli. They can be gram-negative or gram-positive. So you could see gram-negative stained purple. Gram-negative was red or pink. And these are the different shapes. So the coccus, diploid coccus, you can have staphylococcus, streptococcus, you can have bacillus, so diplobacillus, staphylobacillus, streptobacillus, or spirillum, and so on. They can also have an attachment of pili on it. Bacteria can release toxins as well. There are endotoxins and exotoxins. So we'll look at the exotoxins first. That is a protein that is released during growth. Now, what this protein can do is it can bind to the cell and alter function and cause damage to cell membrane. It can enter the cell and cause this damage. Endotoxins come from the lipopolysaccharide in the gram-negative walls. The action is pro-inflammatory. It causes lots of inflammation in the host body. It can be released during bacterial breaking of that cell membrane because the LPS is in the cell membrane and it's getting destroyed, or it can be released during cell growth. So just think about that. If you have somebody that has a bacteria that's probably a gram-negative bacteria, and we give them antibiotics to try and help them with this infection, we end up lysing and destroying the bacterial membrane and destroying the LPS and releasing a ton of pro-inflammatory mediators. So a fungal infection is with eukaryotic organisms, so meaning a true nucleus, and they have a different wall than bacteria. The types include mold, yeast, and dimorphic. Mycosis is an infection with fungus. So an example would be blastomycosis. There's also pneumocystis carinii, which has been reclassified as P. durovici. So here are some fun fungal facts here. They reside on the skin and the mucous membranes. Most require a cooler body temperature, so most are on the surface. Antibiotics do not penetrate the wall, and so you need an antifungal agent. It can cause an opportunistic infection. So an example could be a yeast infection with antibiotics. There are also parasites and protozoa. A protozoa is a unicellular organism such as malaria, amoebic dysentery, or giardia. There are also parasites like helminths or worms, such as roundworms, tapeworms, or flukes. And you can have a parasite such as malaria. Plasmodium, the malarial parasite, is transmitted by anopheles mosquito. It invades the liver cells. The liver cells then rupture and release into the blood. Our red blood cells can get infected, and the plasmodium can multiply within the red blood cells. Signs and symptoms include intermittent fevers, chills, a decrease in your blood count, so anemia, as well as neurological damage and death. So these parasites and bacteria and viruses, they can overcome our host defenses by having microbial adhesion, so it sticks. They can create biofilm formation, which will protect them. They can replicate fast. They can be encapsulated, so they can have a thick carbohydrate and protein coat on them. They can mutate and have different antigenic variations so that they can evade our immune system. They can produce enzymes and toxins that will dissolve our extracellular matrix, and it can also prevent antibiotics from reaching them. They also have the ability to survive within a cell, and they can also coat themselves with the host's self-antigens. So there's a few stages whenever we have an infectious disease. First, we're going to have that incubation period where it's kind of just growing, thinking it as an incubator. And then we're going to have that prodromal phase where we might have small or slight not right feelings. So it's subclinical. You might not have a lot of symptoms. Then you have the acute stage where you do have symptoms, and the convalescent stage when you're getting better. You may have atypical courses of an infection that could include fulminant, which is severe reaction, and you may not have a prodromal effect. You could also have insidious, where it's a prolonged prodromal effect. You could also have subacute or subclinical syndrome, where you may not show any clinical signs or symptoms.